It's really dear to my heart. And also, I want to tell you a little bit about what's on the horizon, what we're going to do with the project that's called Salsa, a project that Brad and his graduate student Ryans and other are involved in. I thought of introducing the topic a little bit by telling you a little bit about my pathway into polar science. This sort of happened as a graduate student when I became addicted to Antarctica. This is the cover of Nature on the summer before I started graduate school. I went to Ohio State actually to study thermophiles. I can tell you that change over a beer sometime. But this paper was meaningful because in the 70s, using techniques, radio echo sounding, there were actually about 70 lakes that were described underneath the ice sheet that pretty much went unnoticed by everyone, including anthropologists who might have thought there would have been the possibility that life could have existed in these ecosystems. The main reason why there wasn't a lot of excitement about that possibility is that this was viewed as an environment that could support life. The view of life in Antarctica at the time was, the biology of Antarctica had been studied for over 100 years, but just the things that swim around the margins. And there are some weird places in Antarctica where there isn't ice. So dry valleys where some mosses and bacteria and nematodes are clinging on to life in the soils in the lakes there. But what is the essence of Antarctica, 97% of what covers the continent, ice, was viewed as an environment that could not support life. In several decades, we now look at Antarctica as going from a lifeless continent to being the largest wetland on the planet, which is a really amazing transformation. This image that you see would be what Antarctica would look like if we could magically lift up that ice sheet and see what's underneath that average of 3 to 4 kilometers of ice that exists there. Everywhere that you see a little dot here, this represents a lake that has been identified. These little lines that you see connecting the lakes and going, these things that look like rivers are actually rivers. We now understand that Antarctica has a hydrological system similar to any other continent. It's just that it's been one that's taken time for us to understand. One of the lakes that really initiated these systems moving from a curiosity to a scientific focus is a lake called Lake Vostok, which you see here. Lake Vostok, this is a big lake. It's about similar in surface area to Lake Erie. By volume, it's the 5th largest lake on the planet. And we just learned that it really existed in the 1990s. What you see here, this is an image of Lake Vostok from space. You're not actually seeing the lake. You're seeing the lake is actually 4 kilometers underneath the surface. What you're seeing is the ice as it flows over the lake, it actually floats. This is kind of like an interior ice shelf. And this is a big lake. It's about 250 kilometers in length, about 50 kilometers in width. If you're a boat in the middle of this lake, you wouldn't see the shores. Now, other than it being a big lake and easy to identify, the other reason why this spurred interest in subglacial lakes is due to the fact that we actually had samples of this material that were available for analysis back in the 90s, in the late 90s. And this is because the lake was actually drilled into much later, but at the time, the Russians built a station over the lake in the late 50s. They actually didn't know that there was a lake there. They just happened to build a station over the largest subglacial lake in Antarctica. And so a core had been drilled through the surface of the ice, and that core had contained a 420,000-year record, paleoclimatic record. And that was the longest and oldest record, ice core record that existed for some time. Now, when they actually started drilling, recovering ice from the very bottom of this, they found ice that had a very different physical and chemical composition, ice crystals 1 meter in diameter. It became quite clear that this ice had a very different origin, that this ice actually represents water from the lake that had frozen onto the bottom of the ice sheet. And the thing about this is what you're seeing here is the flow path through the borehole line. Now, keep in mind that the ice sheets in Antarctica are moving. And as this ice sheet moves and this material accumulates, what you're essentially getting is a transect of samples from the shoreline into the middle of the lake. And so this was material that when I was a graduate student and later working with John Priskew, that was really our first picture or window into what the limnological and biological conditions may be like in these lakes. As I told you, this is a big lake. It's deep. It has two basins, a northern and southern basin. The southern basin has probably got 100 meters of water column underneath the ice, as well as likely hundreds of meters of sediments that underlie that. Now, we didn't have access to the lake at the time, but one of the questions that we were trying to address, these lakes are different than lakes on the surface in that there's obviously no sunlight here. So as a microbioecologist, we're interested in what the microbes eat. Where are they getting their energy and new carbon from? And one possibility is that they could actually be getting this material from the ice. Now, the reason that there's a lake here is that when you put ice over thick ice, even in a place like Vostok Station where the surface temperature probably a day like today is minus 90, you're trapping geothermal heat. And if the ice is thick enough, that ice at the base will actually be at the pressure melting point. So this water, the reason that there's water here is this is melting. The ice is melting at the base. Now this slant in the ice ceiling is just not my poor artistic skills, but this is a real thing. This slant, because this ice is deeper, it is melting. This ice, which is shallower in the southern portion, is actually colder. So this means that while ice in this portion of Lake Vostok is melting in, this is where the accretion is happening. So, this, we can do things because we were able to characterize what's in the ice, what's in the accretion ice. It allows us to sort of make, to constrain the amount of dissolved organic carbon that is entering the lake through the ice. So it's possible that organic carbon that's entrapped and introduced in the lake could actually support the biota of this lake. And what we did by characterizing the amount of DOC that's entering through melting ice, as well as that's being removed through accretion, and making some assumptions, some of which are almost certainly not true, we can come up with a rate that could actually feed the organisms in the lake. And this number may mean nothing to you, but what I can tell you that this is about 2 orders of magnitude lower of carbon throughput that we could expect that would maintain reproductive growth. So in other words, if organisms in the lake are subsisting on these materials, they're probably in some kind of state of maintenance or something. Not exactly an exciting scenario, an ecological scenario for a lake. But of course, what we didn't know at the time, because we couldn't enter it, is there were a lot of other sources of DOC that we couldn't constrain, specifically those scoured into the lake and those that would be in the deep sediments. There's also a possibility that what could replace the photosynthetic component of these ecosystems that are on the surface is actually organisms that are chemoautotrophs. And so there's actually some evidence in the accretion ice that there may be geothermal input to the lake. And of course, if this is true, it could mean that the deep waters of Lake Vostok, could harbor communities similar to those found around hydrothermal vents. You don't need to invoke geothermal input into a lake, though, to really, for a scenario where chemosynthesis may be possible. For example, this ice sheet, as I described, is moving. And it is moving over bedrock. That comminution of that bedrock is being bulldozed in the lake. And that introduces minerals like pyrite, which can serve as potential electron donors either in the presence or absence of oxygen. And these could be mechanisms that are driving essentially dark primary production in these ecosystems. And actually in studies of the accretion ice, we found evidence for organisms that could potentially carry out this process. Other sources of energy include methane in the sediments. Of course, at that time, we were not able to characterize the water in any depth. Now, our conceptual understanding of the hydrology of Antarctica took a complete turn in 2006 as a result of data published from a satellite called ICESat. Some of you may have known that ICESat-2 was launched in the last couple of weeks. So this is the prerequisite to ICESat-2. ICESat was a is a satellite that's equipped with an instrument called a laser altimeter. And what it is the satellite to do is to make very precise measurements to measure the surface of the ice sheet. What unexpectedly these surveys found in Antarctica, that there were regions of the ice sheet that were moving up and down meters, 4 to 5, up to 10 meters in a period of months. There is no mechanism to add ice to the base of an ice sheet. The way you remove it is melting. But even if you set off a nuclear bomb underneath the ice sheet, it could not remove this. So the explanation for this is that these displacements are actually, these movements in the ice sheet are actually due to water moving underneath the ice sheet. And it's this combined with the work on the evidence for biology occurring in these lakes that really motivated a project called WIZARD. WIZARD was an overarching project that included not just teams of microbiologists and geochemists, which I want to introduce in just a moment, those that were interested in the effect that water, and more specifically water moving in large volumes underneath the ice sheet, how that affects movement, as well as the stability of this region. This area of West Antarctica is looked at many like a time bomb. It sits all underneath sea level. And so there's a lot of interest in its stability and how it's reacted in the past. Now the data I'm going to be talking about today was gathered by a collective effort of this group. PIs, myself, John Priskew, Mark Skidmore, and 90% of the data that I'm going to show my students. Amanda Ockberter and Peyton Atkins, my students, Alex Michaud and Tristy Vic Majors. These are John Priskew students. Just to give you a little idea of what is involved in an effort like this, we're going into the field with large amounts of equipment to drill through the ice sheet, as well as laboratories that we have right on site, so the materials coming up the borehole, we can conduct analyses that cannot wait. Now our study site for WIZARD and for the Salsa project is the Seiple Gould Coast of Antarctica, and this just on this locator map gives you an idea where this is. What you see here, this black line on this map, represents the grounding line. So everything to the right of this is actually seawater. The ice sheet is grounded on this portion on the left upward. This point right here, that's the most southern reach of the ocean, just so you now know. Every place you see a little yellow outline is a lake. We think of this as a lake district in this region of Antarctica. And in 2013, so data I'm going to tell you about, we drilled into subglacial Lake Whillans. I talked to you about how these lakes were identified by the way they move the ice sheet up and down as they fill in flood. What you're actually seeing here is a plot of ice sheet elevation changes over about a decade. This lake has filled and drained about 3 times over this period of time. You'll note that this blue line here represents the pathway of water that comes from upstream and enters the lake and then eventually flows down and flows into the Ross Sea. Now, of course, water moving through this drainage system has never been directly observed. This is all curling subglacially and is inferred by the movements of the ice sheet on the surface. As a part of WIZARD, we also drilled into a region where we knew that the water exited into the Ross Sea, and I'll show you a little bit from that project as well. As part of Salsa, where we'll be deploying in January and drilling through another lake. This lake is about 100 kilometers or so from subglacial Lake Whillans. It also receives inflow from the Mercer ice stream. We actually know some of the water is coming from East Antarctica, so the source of water ending in the lake is a little different. It's also a little more distant from the grounding zone, which may be reflected in the nature of the sediments, which are preserved there. And I know this is really hard to see in the back, but this is actually an updated surface elevation profile of the lake that we just got. This is up into 2018. So you can see when we drill it this year, it's probably going to be filled. And this lake moves up and down about 12 meters. So we think that there's at least 12 meters of water there, but maybe there's 50 meters. We actually, it's a little difficult to actually. We know that there's water there. How much water is there? Probably not more than 100 meters, but. Now, there's a paper published in Nature a month or two ago, which actually used some cores which were collected from the study. This is subglacial Lake Whillans and the grounding zone, as well as cores that had been recovered in the 90s from upper portions of the ice stream. And so using C14 dates that were obtained from these cores, in addition to ice sheet modeling, they were able to show, well, first of all, at the last glacial maximum, this is the predicted, this is where the grounding light is predicted to exist. So it's just sort of thought, this is here, this is its current location. So conventional wisdom is that this is just gradually receded over the last 20,000 years. But what this study found is that the ice sheet actually retreated back into a region, including subglacial Whillans. It's not on this map, but Mercer is right about here. This means that about 10,000 years ago, that the systems that we're drilling into might have looked like this. These are video that we captured from at the grounding zone during Wizard. This is open ocean water. We think that the water actually takes about a decade to get here, but really circulation patterns on the Ross Ice Shelf are not very well known. So it wasn't really surprised to find fish in the ocean, but we wonder what they eat this far away from open water. But the main point here is that these freshwater lakes that we're studying today in the very recent past were actually connected to the ocean. And it has lots of implications on how they may operate, which I'll expand on here in just a moment. Just to get an idea what it looks like when you go car camping with 1 million pounds, this is what it looks like. We have about 50 people in the field for 3 weeks during the field season. We are the second largest settlement on the Antarctic continent. This is really the business end of the operation. Each one of these that you see is a 40-foot container. Each of those is filled with a hot water heater. Essentially what you'd see at your car wash, we have 3 of these in each container. These are taking water and heating it up. We are using hot water to actually drill through the ice sheet, and I'll come to why we do that in just a moment. I mentioned labs. We have 2 field labs, one primarily a sediment lab, one a biology lab, fully outfitted. Incredible resource to have right there in the field. This is our borehole right here in our hose wheel and our major operations. One of the things we're concerned about, in fact, we spent about 3 years figuring out how we would do it, is that we needed to drill into the lake in a way that wouldn't disturb it. It's not just for reasons of environmental protection. It's certainly a strong motivation. But if we drill into the lake and contaminate it and then we collect our samples, they're worthless. So our strategy was to use hot water that we cleaned in ways that I'll describe in just a moment. And we drilled to about 700 meters. We just have, basically we're doing this with a 1 kilometer garden hose. We don't have fancy sensors or anything on the end of us that can really tell us, hey, you're in the lake or you're this deep. We don't have that kind of information. So at 700 meters, we stopped, we pulled out the drill, and we did a hydrocast, and I'll show you some data from this in a moment. Before we initiated drilling, again, what we did is we removed about 30 meters of water from the borehole. And this is important because this hydrostatic imbalance means that when we actually drilled in the lake, instead of the borehole entering the lake entered the borehole. I'm telling you, we don't have fancy sensors. The only way that we know this happens is we have a pressure transducer in the borehole. So what you see here is going on and whoop, all of a sudden the water level goes up 30 meters. Now we know we're in the lake. We did CTD cast into the borehole lake. I think the only data that you're seeing here from the CTD cast are the temperature. These measurements were based on discrete hydrocast in the borehole and in the lake. What you can see from the conductivity and the pH is that we're sampling two distinct water masses. When we look at the concentration of cells that we find in the borehole, cells and biomass, there's about 200 times more cells in the lake than there are on the borehole, and about 100 times more biomass. So this is a scenario that gave us confidence, and we're actually getting water from the lake, that we're collecting organisms for there, not things that we've introduced or not things that have just been drug in the borehole water. The lake was undersaturated with oxygen. Some of you may be surprised that there's oxygen in the lake at all. I told you that the reason there's water here is because the ice melts. The ice, by volume, is about 10% gas. That's atmospheric gas, and 20% of that is oxygen. Undersaturated with oxygen, relatively, surprisingly high levels of DOC. This is about 4 times the levels you might see in the open ocean. Based on nitrogen to phosphorus ratios, the system was limited in nitrogen with respect to phosphorus. About 3/4 of that inorganic nitrogen was was ammonium. And that actually raised the possibility that that high level ammonium could actually be driving a chemolitho-autotrophic process called nitrification. And indeed, when we look at the CAP 17O of nitrate, the values that we get are consistent with its production not being from atmospheric nitrate, but production through nitrification. You saw the large differences in the conductivity between the borehole and the lake. The ice is essentially distilled water. I mean, Antarctic ice is one of the purest substances on the planet. So where are the solutes coming from? Certainly the water is interacting with the sediments, and there could be weathering component, but also I just talked about its connection to marine sources, so there could be a marine component as well. If we look at conserved species like chloride and look at the ratio specifically of bromine to chloride, we see that their ratio in subglacial lake water is very similar to what you find in seawater. And so by using chloride, you can use the chloride, the amount of chloride and the ratio of the other cations and ions in seawater to actually estimate the seawater component of the solute source, as well as the component that can't be explained by seawater. And so this is what we infer to be the weathering component. We can explain a number of these solutes from various weathering patterns, various weathering reactions. Magnesium is not negative. This is probably a result of ion exchange with like clays in the sediment. This has actually been supported by some experiments one of Mark Skidmore's students have done. This lake is not contemporarily exchanging with the Ross Sea. This is about 100 kilometers, so it's not like at high tide that the ocean comes into the lake. That kind of exchange only happens within about 1 kilometer of the grounding zone. Where is this seawater coming from? Well, if you actually look at the concentration of chloride in the lake and then look at it in pore waters, so these are data from multicores that we pulled, if you're down at about 35 centimeters, the lake water is about 0.5% seawater, and when you get down to about 35 centimeters, it's about 4% seawater. So clearly, this is coming from the sediments. And if you look at the delta 18O of the pore water at 35 centimeters, it's minus 36.5. And if you take 4% seawater and mix that with glacial melt at minus 38, you get the same number. One of the first measurements that we did on water that came up in the borehole is we had a microscope in the lab, and within an hour of the water coming up, we were looking at images like you see here in A. This is an epifluorescence microscopic images. So these cells are green because we're treating them with a dye that allows them to fluoresce. them with a specific wavelength of light. Now, of course, just looking at these green dots and filaments doesn't tell us what the organism is, but the reason that this was a high five moment was because there were so many cells there, cells that must be eating something. What the images you see here, these are just some scanning electron micrographs, which show some of the various cell morphologies that we encountered in the lake. Now, I said that we spent a lot of time trying to figure out how we enter this lake cleanly, and that actually involved building a water purification system, which consisted of large volume filtration units which could pass water through them through 0.2 micron filter at 100 gallons per minute. That water was then subsequently hit with high intensity UV light. We have about 1 kilometer of hose and wire that we have to put in the borehole every time we put in an instrument, and that stuff is filthy. Our solution to that is, well, of course, all instruments that go down there, we can easily just spray with 3% hydrogen peroxide, which degrades into hydrogen and oxygen, sort of leave no trace. And everything that went through this very high intensity UV borehole collar. Essentially, passing through this collar at 1 meter per second can do a 6 log reduction in bacterial endospores, one of the toughest things that we know. So this is actually, as I've been telling Ryan, and this is probably the scariest element of the project. Now, we're able to collect samples from the system, water that was entering the borehole, water that was leaving. This is very easy for us to do on the surface. We did hydrocast within the borehole before we entered it, and of course we sampled the lake and the sediment. And what you see here on the right is an NMS deordination of data that we obtained from analyzing the composition of these communities. And we're just using these are sort of the same techniques that are used in the human microbiome. We're amplifying a gene that all organisms have, and we're using that to infer community structure. Now, I'm going to tell you a little bit more about those communities in a moment, but the point I'd like to make here is that those communities that we found in the drilling water were significantly different from anything that was found in the lake and the sediment. And so the data that I'm going to be able to tell you about, the microbiological aspect, and this is data that's coming from the lake. These are not things that we drug in. Now, we were able to collect water using a system which we lower in the lake, and there's a 3-tiered system here which actually fractionates the water into 3 different size classes. Hopefully you can see this. We're collecting material on 3.8 and 0.2 micron filters, as well as shallow cores. We were only able to do our about half a meter. Brad's going to core down to, what, 75 meters is how long this core is going to be? Okay. So the techniques, the data that I'm showing here are based on the very standard sort of 16S ribosomal RNA molecule, as well as our approach where we're taking all the DNA and sequencing it. And so what you're seeing is that the lake was primarily bacterial. There is one group of archaea, that what you're seeing here, proteobacteria, actinobacteria, and one group of archaea, the Talm archaea, that I'll tell you a little bit about in a second. When we first were looking at the samples, we were using techniques that target eukarya, and we were coming up with nothing. So these are primer-based methods. People who do this will know what I'm talking about. We just thought maybe we had terrible primers. We didn't expect there to be fish, or maybe a nematode in the system, but certainly microbial eukaryotes, fungi were things that we expected to see. It wasn't until we got back the metagenomic data, spelled wrong here, where we found very low evidence for eukaryotic DNA in the libraries. We were surprised by this. This really seems to be a system that is, this level, by the way, that's probably the same amount of human DNA is in these. That's consistent with a level of contamination. What else I want to point out here is that we see that there are clear trends in distributions between, as we go into the sediment. We have this major beta proteobacteria, or big hitters, in this system. As we go with depth in the sediment, that changes. This just gives you a little better idea Yeah, what I'm showing here are rank distributions of the species that we detected in the water column, and these are the superficial sediments. If you look at the things that are most abundant in the system, what is shown here and in blue and red, I've summarized over here in this tree. A couple things come to mind. What we're doing here, by the way, for those, I should remind myself to say this, this is a hypothesis generator. We're obtaining DNA sequences, and then we're looking to see what those sequences are related to, and we're generating hypotheses on what they may do, all right? Something that came to our attention, especially in the water column, high representation of species that affiliate with Nitrotoga and Nitrosa archeum. These are two groups which are known to participate in a process called nitrification, a lithotrophic process. We also found good evidence for organisms that are related to species like sideroxidans and Thiobacillus denitrificans. These are also chemolithoautotrophic species which can use reduced sulfur or iron compounds to fuel their metabolism. One other thing of note, a group that we found not very abundant in the water column, but very abundant in the surface sediments, are a group called anatrophs. So there's another compound besides inorganic elements that could add new energy to the system. If there's methane percolating up from these sediments, these groups could actually be oxidizing and not just getting energy from themselves, but adding new carbon to the system that other organisms could use. I also wanted to point out that we found that what the communities were on the very smallest pore sizes that we analyzed were pretty close. We did see some larger differences in greater than 0.3 and 10 fraction. One of the things we noticed is that some taxa, which are actually associated with iron oxidizing bacteria, were present in the larger fractions. And this could be explained by the fact that these might be big cells, but more probably, these are probably cells that are attached to solid particulates, probably particulates where they may be gaining that iron from that were present in the water column. And I think I showed pictures, but I may not explicitly point it out. Those are a lot of particles in this water, very small little particles. All right. The way to demonstrate autotrophy is actually to measure it. And we're doing that by how you would measure primary production in every other environment, but we're just not putting them in the light. So these are values of dark CO2 fixation, which are on the order of about 33 nanograms of carbon per liter today. So that's the amount of new carbon that we can measure that's coming into the system. We don't specifically know what processes are doing that from this data, but if we eliminate the fact, we know that some of that carbon is being burned off by heterotrophs like us, and when we account for respiration, we're surprisingly finding net accumulation of organic material based on these measurements. Now, to get some information on what organisms may be doing this, we lean on the metagenomic data. And what that seems to indicate is that the predominant carbon fixation genes are associated with the Calvin cycle, and most predominant in organisms that are living in the small are actually associated with smaller particles in the water column and in the superficial sediments. These carbon fixation genes seem to drop off as we go with depth in the sediment. So the organisms that are likely to be doing this, in other words, the chemolithoautotrophic pathways that we find that are most abundant in the metagenomic data, are associated with organisms that can either use reduced sulfite compounds or, again, ammonia oxidation. This is also consistent with the isotopic data indicating that the nitrate in the lake is not atmospheric in source, but is coming from nitrification. Now, there has been a lot of speculation about the fact that there could be huge reservoirs of methane just sitting underneath the Antarctic ice sheet. And the question has emerged, what happens as these ice sheets retreat? Is it actually possible that this could be associated with huge amounts of methane release to the atmosphere, which could lead to a positive feedback that would lead to further warming. Well, there is the estimates of the amount of organic carbon on the Antarctic ice sheet, 21,000 petagrams. If you mineralize that to methane, yes, that could make a difference. The thing is, we never had any, there were no data available for methane under Antarctica. These are the first data that we've been able to obtain. What you're seeing here in black, this is concentration of methane with depth down to 38 centimeters. You'll note that these data, they're a little bit noisy, and it has to do with the fact that when we bring up the cores, we have no way to keep them pressurized when we're sampling. I hope you can see this, but you actually see the degassing going off. So probably these are conservative estimates for how much methane is actually in the system. But we're somewhere in the range of 250 to 300 micromolar methane in the deepest portions of this core. And if you look at the stable isotopic composition of the carbon in that methane, this is consistent with it being produced through hydrogen-trophic methanogenesis. We don't know whether or not this is actually being made contemporarily by methanogens that are in the sediment. When we actually look for methanogens, the deepest depth that we went to, well, we find some, but if you look, they're just 0.1, but they're not very abundant. This could be methane that has been stored in the sediments. Deeper cores are actually going to help with this. The other thing I want to point out is the concentration of methane in the water column is basically air equilibrated water. So almost all this methane is being consumed probably right here at that sediment interface, where the methanotrophs are most abundant in the system. So I'm not saying that these organisms are saving the world, but the idea that you could have methane release is still out there, but methanotrophy is something that could actually attenuate this response. And we've actually seen very similar things in Greenland as well. So this has allowed us to, this data has allowed us to build a sort of conceptual model of how we understand this lake to operate. The major driver in this system is processes that are going on in the sediment. I don't just mean the sediment community, but reactions, weathering reactions, biological reactions, which are feeding processes in the aerobic waters. So the sediment, as I said, the organic matter in there is most certainly of marine origin, and in fact of probably recent marine origin within the last 10,000 years. The mineralization of that material in the sediments to ammonia and methane are feeding processes in the water column like nitrification, like methanotrophy, which are contributing new carbon to the system. When you combine those two, it's roughly 65 nanograms of carbon per liter a day that are entering through this process. We also have evidence that reduced compounds like ferrous iron and sulfide could be driving chemolithotrophy. Now, one of the things that's sort of unusual about these sediments, which have plenty of sulfate in them and are of marine origin, we might really expect to be sulfate reduction to be gangbusters in these sediments. These sediments weren't sulfitic. The sulfate profiles are like this. We can actually find sulfate reducers at some frequency in the communities. Take them, you can amend them with lactate, and you can measure a little bit of cryptic sulfate reduction. Sulfate reduction does not seem to be a predominant process in here. It sort of makes us wonder where the sulfide is coming from. And we think that that can be explained through these sort of pyrite weathering reactions that I mentioned very early in my talk. Thiosulfate is sort of a metastable intermediate in the process, so it's possible the reduced sulfur is not coming from sulfate reduction in this system. We had very weak evidence for it occurring. So this brings us to, this sort of sets the stage for Salsa. As Brad mentioned, this project will look into a lake that originally we were to look at two lakes. Brad, do you remember this? I don't know if you've already forgotten, but the proposal actually said we'd look at two lakes. Subglacial Lake Ellsworth, which is right here. It's not Ellsworth, it's Ellington. Engelhart, yes. It's in one of the most heavily crevassed areas on the ice sheet, so that was actually thrown out by Antarctic Logistics. But here's our right here. You can see that our overarching hypothesis is tying into this idea. This connection of these systems to the marine environment is clearly important in the biology we observed in subglacial Lake Whillans. How does the biology of this system compare to others, the other 398 lakes that exist in Antarctica? Salsa will not answer that question, but my sense is, and I think that these lakes might be the tropical rainforests of the subglacial world, in that the interaction with the ocean is providing a lot of energy into these systems. Whereas a lake like Lake Vostok, isolated, it hasn't seen the surface for 15 million years. There may be a very different kind of biology going on there. I'll just point out that if you're really interested in following us, and I know this group is, to see what Brad is up to during January and February, you can follow us on social media. The last thing that I wanted to mention is that the systems under the ice in Antarctica are actually serving as benchmarks to try to understand processes that are going on elsewhere. When I was a graduate student in the late 90s, there was a lot of excitement about the fact that Europa could have an ocean underneath its ice shell. We now have a lot of confidence that there's actually an ocean there. So in that short period of time, not only did we find out that Earth is not the only place that has an ocean, it's not even the biggest ocean. There are 12 worlds in our solar systems that have oceans identified beneath them. Now they're below ice. In about 5 years, there will be a remote sensing mission sent to Europa called Clipper. And that will do a lot of kind of the measurements that on Europa that was done back in the 70s in Antarctica when these lakes were first described. What you may not know is that there's a potential mission that has been slated. I was actually fortunate enough in 2017 to sit on the science definition team for this project. This is a lander that would land on the surface of Europa. It would drill into the near surface, probably no more than 10 centimeters to a meter, for the sole purpose of trying to find evidence for life. This is a historic opportunity in that the last time NASA had a mission like this was in the 70s, Viking. Viking is the last time that NASA actually created a mission to specifically look for life. And if this thing actually goes through, in 10 years, we may be looking at data coming from Europa. And the encouraging news is that the next step of this is actually building instruments that you would put on this payload that would make this hull. And in August, that happened, in that the program was launched, proposals were, now this could be canceled at any time, but it is I believe that it's an exciting time ahead in Antarctica and elsewhere for life, watery life under ice. So with that, I'll just end with a few comments. And since I was a graduate student, it has been such a transformation in our understanding of the habitability of places not like Antarctica and how that has sort of transformed our understanding of the possibility that you can go to a place, a cold place like Europa, and still plausibly think that there could be life there. Lakes like Vostok were viewed as sort of these stagnant bodies that, stagnant and not mixing sounds like not very exciting from a point of view for energy flow in organisms. But the idea that we have exchange of organisms, energy, active hydrology, that's really changed the way we thought about life. And also, every ice model that's out there that tries to predict how the ice behave, they rely on the fact that the ice is frozen to the base. When you get water, and you got lots of water moving rapidly, that changes the game. We've looked in one place so far. After February, we've looked in 2 places in pretty much the same region. The amount of water underneath the ice sheet, if you put it on the surface, it would be a layer 2 meters thick. I will be shocked if life is not ubiquitous in all those aquatic environments. And this is, an area about we've ignored it. Its biogeochemical contribution, we haven't even thought about it. And it's kind of like it would be like ignoring Russia and saying, oh, we just won't count that in a biogeochemical model. How do they make their living? Any way they can. The organisms, if there's organic carbon in the sediment that's being transformed, they will use it. And you can also have rock eaters. The physical action of the ice sheet, grinding rock, putting it in solution with water, that seems to be the ticket. The idea that there could be huge volumes of methane housed, we've collected the first data of methane. It's there. Not surprisingly. Take microbes and organic carbon, take away oxygen, they'll turn it into methane. The important thing is that if microbes are actually causing this problem, they might be partially solving it too. And that when you add oxygen into this water, they can actually attenuate that response through a process called methanotrophy. And just another thing that maybe we haven't thought about a lot is that this active hydrology, microbes mobilizing elements from the lithosphere into the Southern Ocean, mobilizing elements like iron into an area of the ocean. A lot of areas of the Southern Ocean are iron limited. How does that affect the biological and chemical systems in the Southern Ocean? I'd say that's a question to address in the future. So with that, I will pause and do my best to answer any questions you may have. Thank you.